Okay, well, let me introduce myself. So my name's Peter Karp, and I work at a research institute called SRI that's about a mile away. It's over by the Menlo Park train station. We used to be part of Stanford. SRI used to be Stanford Research Institute, but for those of you who don't know, we became independent from Stanford in the 1970s, and the SRI doesn't stand for anything anymore. It's just SRI International. I did my Ph.D. here at Stanford in the 1980s in computer science. Great. Well, let's launch into the talk then. Here's a quick overview of the talk. I'm going to give you an overview of our BioCyc database collection and pathway tool software. These are tools that my group has been working on since the early 1990s, so 25 years or so. And then after giving a general overview, I'm going to focus in on this talk on various visualization capabilities that we've built into the software over the years. Well, BioCyc is a database collection. It's a collection of about 11,000 what we call pathway genome databases. And we chose that name because these databases have pretty broad schemas that integrate many different types of information. Information about the metabolic network of an organism, its metabolic pathways, reactions, and substrates, and then the proteome of the organism, and the genome of the organism, and the genetic regulatory network of the organism. Although for the last part, the genetic regulatory network, that's only really present for a handful of the organisms. And we divide these 11,000 databases into 3 tiers to try to communicate the amount of curation that's gone into the databases. So the tier 1 databases, of which there are roughly 5, are highly curated databases, like our E. coli and MetaCyc databases both have more than 2 person decades of curation that have gone into them. That is manual extraction of information from the biomedical literature. The tier 2 databases have typically received somewhere under a person year of curation. And then the tier 3 databases, which are by far the most common, have undergone no curation at all. They're purely computationally generated. And overall, these databases, the tier 1 and tier 2 databases have been curated from a total of 66,000 different publications. That's the number of publications that our curators have extracted information from. Well, the Pathway Tool software is the software that we use to generate the tier 3 databases. The software contains interactive editing tools that our curators use to do their curation. And then there's a whole range of other different tools within Pathway Tools for querying and visualizing the contents of these databases. So one use case of Pathway Tools is to create and maintain over time a curated database about one organism, about its genome and metabolic pathways and so forth. It also allows one to generate a quantitative metabolic flux model from one of these databases. There are various omics data or large-scale data interpretation tools present in Pathway Tools. There are comparative analysis tools for comparing information across different organisms. And I mean, one thing I guess I should say is that the data in these organisms in these databases is coming from sequenced genomes, ultimately. So people in the scientific community will sequence a bacterial genome or a plant genome. They'll deposit it in the GenBank database, and then we extract it from the GenBank database and then do various value-added operations on the data, which I'll give you an overview of in a minute. So Pathway Tools is about 700,000 lines of code, of which the majority is Common Lisp. There's a little SQL code. There's probably, 20 or 30,000 lines of JavaScript code. We've been doing more and more JavaScript lately on the user interface side, but the vast majority of the code is Common Lisp. And we've chosen Common Lisp because everybody in my group likes working in Common Lisp. Because you can do things faster with less attention to mundane details of programming, and because we can maintain this huge code base with a relatively small number of people. Because Lisp is such a high-level language, we can get new things working more efficiently than you can in other languages. We can debug them more effectively. We can maintain the code more effectively. We have a group of about 8 programmers who are working on the software now. That's been the size of my group for at least 10 years now. And the software has been licensed by more than 8,000 groups. And one of the common use cases for these groups is they'll do one of the same things that we do with the software is they'll have sequenced some genome in their lab and they'll want to analyze it, write a paper about it. And one of the things they do in the course of that analysis, they'll run a number of different software tools across their genome that they've sequenced, including our software. And one of the capabilities that our software has is predicting the metabolic network of the organism. Any questions about this high-level overview? Feel free to ask questions since we got a small group here. So in more detail, how do we actually create a BioCyc database? Well, this is the basic set of operations that happen. We start by obtaining a sequenced genome from the NIH GenBank database, or more specifically, the RefSeq portion of GenBank. And so we'll download not only the DNA sequence, but also what's called the annotation of the genome, which means there's already been a fair amount of attention paid to running various computer programs and interpreting their results to predict where are the genes within that genome and what are the functions of those genes. Okay, and so the annotated genome contains not only the DNA sequence, but the coordinates of these predicted genes and the predicted functions of these genes. And then that's where our software comes into play, is we then run a combination of computational inference tools within Pathway Tools, and we do a series of data import operations where we're importing data about this same organism from a variety of other databases that other groups around the world maintain. And so one thing we do, our software does, is we predict what reactions the enzymes coded by the genome catalyze. We predict both metabolic reactions and transport events. We also predict the organization of those metabolic reactions into larger groupings called metabolic pathways. And we predict some things called pathway hole fillers and operons, or clusters of genes that are regulated as a unit. And we compute orthologs across these databases, or essentially equivalent genes in different organisms. And we compute PFAM domains, if you know what those are. And then in terms of importing data, we're importing data from various other databases where there are other groups that collect information that's basically complementary to the information that our curators collect. And this includes regulatory data, genetic regulatory information, the subcellular locations of different proteins, features on different proteins, like where their active sites or metal ion binding sites, gene essentiality data, because there are more and more large-scale data sets now where people experimentally knock out genes in different organisms and determine if those knockouts are lethal or not. We import those data sets as well. And data called phenotype microarray data, which basically is more high-throughput data that describes whether these different organisms can grow on all sorts of different chemical conditions. Do they show growth or no growth? So we basically have a pretty automated pipeline for doing all these different things. And then if finances allow, we send the database to our curators for further curation. That's pretty rare for most databases. So most databases then just go into BioCyc without any curation. And one of the things that can happen during curation is to develop a quantitative metabolic flux model that's able to make predictions about which reactions are carrying how much flux during the operation of the metabolic network when the organism is growing on different nutrients. Well, the very first database in BioCyc of the 11,000 was EcoCyc. It's an E. coli database. And that project started in about 1992. And We've had a group of people working on extracting information from the literature about the functions of E. coli genes and E. coli metabolic pathways and regulation of E. coli genes by various mechanisms. And over the years, they've extracted information from 33,000 different publications. And we know that because the database cites those 33,000 publications. So when we extract information, the database will cite the source of that information. And these are among the different types of information that are present in EcoCyc, and we've discussed a lot of this already. And so the curators, they kind of do two kinds of things when they're extracting information from a publication. They'll take little atomic pieces of information and put them into appropriate fields of the database, like the molecular weight of an enzyme, or the PI of an enzyme, or kinetic information about an enzyme. Lots of numbers. But they'll also write what we call mini reviews about a gene. So they'll write 1 or 2 or more paragraphs that summarize what's known about the role this gene plays for a given organism. And so we call those things mini reviews. If we count up all the mini reviews in EcoCyc, it adds up to about 2,300 textbook page equivalents is the amount of information that our curators have authored over the years. And we publish an article on this database every 2 years or so in the Nucleic Acids Research Database issue. And here's a schematic of how many objects of these different types are present in the database. So there are 4,500 gene objects. So there's one gene object in the database for each different E. coli gene. And there's an object in the database for each chemical compound that E. coli is known to have an enzyme that acts upon. There's an object in the database for each metabolic reaction, each chemical transformation among these different metabolites that's catalyzed by some E. coli enzyme. And then there's a distinct object for each metabolic pathway in the organism. And so here's one way to look at what Pathway Tools lets us do with one of these pathway genome databases is the collection of information in these databases can be used in several ways. We can generate several different types of information pages on the web about genes or metabolic pathways or metabolic reactions. So that's one use, is to create an online encyclopedia. It's kind of like wiki pages, but they're generated dynamically from the contents of the database rather than being authored by people.
Have you heard of Metabase?
No.
There was a paper published, I think it was in 2012, whose sole purpose was to actually compose a wiki for all sorts of different databases. So they kind of thought of themselves like the database of databases, if you will. And the paper cited like 40 or something so times. I'm not sure if it caught on necessarily, but it is a very interesting effort that I just want to kind of throw out there and maybe talk about after.
Okay. Yeah, I haven't heard of it, but some other uses of the database is that we can generate these zoomable metabolic map diagrams directly from the database. That's one of the visualizations we generate. And that can be used for high throughput data analysis. I'll talk about that shortly. We also have various query tools for letting scientists extract sub-collections of information from the database, like find all proteins that have some properties of interest to the scientist. And we can generate quantitative metabolic models from the database as well. So these are all different parts of Pathway Tools that let us do fairly different things with different subsets of the data in one of these pathway genome databases. So how do scientists use a database like EcoCyc? Well, it's used as an encyclopedia that experimentalists, when they're working on some gene of interest, they'll go to the database to find out what's known about that gene. Another common use is computational biologists or bioinformaticists, when they want to create some new computational predictive tool, like to predict operons in bacteria or promoters in bacteria, they'll need a training set of data. And often they'll extract that training set from EcoCyc because it provides a really well-curated, well-known collection of training data that they can be confident are correct. Also, people who want to redesign organisms' metabolic machinery, like to produce new chemicals, they'll use the database as a reference source on the starting metabolic network of the organism. So the vast majority of databases in BioCyc are like EcoCyc. They're databases that describe one organism whose DNA has been sequenced, and they serve as a knowledge reference source on that organism. But this one database, MetaCyc, is different from all of those other databases in that MetaCyc is kind of a universal metabolic pathway database whose goal is to describe metabolic reactions and pathways and metabolites from all domains of life. So the idea is our curators go out to the literature and extract known reactions and pathways from the literature from all different organisms, all domains of life, plants, bacteria, mammals, everything, and collect them all together to give us this kind of universal collection of pathways. And then that's the reference source of pathways that our pathway prediction tools use when we're analyzing a new genome and predicting what metabolic pathways are present in a new genome. Essentially what the predictive tools do are they kind of compare the set of enzymes in the genome that is being analyzed against the enzymes and reactions in MetaCyc and use them to pull out from MetaCyc the reactions and pathways that are relevant to this organism. So essentially, when we're predicting pathways for a new organism, we're pulling some subset of MetaCyc into our new database to constitute that organism's metabolic network. Okay. If you're familiar with some other databases in the field, like KEGG or SEED, you can see MetaCyc is a good bit larger in most respects. Many more pathways, many more reactions. KEGG does have more chemical compounds than we do. Okay, so a little bit more about pathway tools. So here's a little bit more about the information flow in pathway tools. So we feed in an annotated genome as the input to pathway tools, and the pathologic component of pathway tools takes that annotated genome, generates a new pathway genome database for it, and then runs all these computational inference tools that we saw on that previous slide, like predicting the metabolic network. So we're kind of taking the genome of the organism and then layering all sorts of computationally inferred information on top of the genome to add value to give scientists interested in this organism more knowledge of the organism. Now, of course, not all those computational inferences are correct. They all have a certain error rate, but this is kind of the best that can be done right now. And then there are three other components of pathway tools that will come into play. Curators will use the editor tools to update the database manually. The navigator portion of pathway tools is what scientists use to query the database, visualize the contents of the database. And then the metaflux tool is what's used to generate a new quantitative metabolic model for the organism. Another way of slicing up the different components of pathway tools is this is more an explanation of what tools are present in the navigator. So the navigator has a variety of tools for operating on different data types within a pathway genome database, a PGDB. So there are genome informatics tools that the user can use to search the genome component of the database. So like there's a gene and a protein search or a set of search tools. There's a gene information page that the software will generate. There's a genome browser that lets the user browse through the entire genome of the organism visually. And then there are various sequence search and sequence alignment tools as well. There are pathway informatics tools, which the user can use to search for a metabolic pathway, display an individual pathway diagram using a layout that's generated automatically. The user can search for reactions according to different criteria. They can search for chemical compounds. There's also what's called a metabolic root search tool that lets the user specify a starting and an ending compound, and the tool will search for the shortest path through the metabolic network that connects those two compounds. And then there's a zoomable organism-specific metabolic map diagram that I'll show you in more detail in just a moment. Then there are also tools for analyzing gene expression information and metabolite information that comes from various high-throughput data sets that biologists can generate. And so one example of these tools are what are called enrichment analysis. Like if you have a gene expression data set, these tools can tell you, is your data set overrepresented? Do you have more genes in a specific area of cell function than you'd expect to find by chance? There are also a lot of pathway visualization tools so that you can paint gene expression data onto pathway maps, which I'll show you in a few minutes. Okay, I think I'll skip that one. Okay, let's talk now about some of the visualization tools, which I've kind of mentioned at a high level. So one set of tools are concerned with the regulatory network information within a pathway genome database. And these are the 4 organisms within BioCyc for which we have the most regulatory information. Essentially, regulatory information tells us information about when does the organism express different genes and what biological circuitry inside the organism is responsible for controlling when genes are turned on and when genes are turned off. And so one of the things we've done is to generate a little cartoon diagram that tries to summarize for the user every known regulatory influence on a given gene. And so our objective here overall is there's a huge amount of information packed into these databases, and what we're trying to do is to generate tools that let the user absorb this information as quickly as possible. It's pretty inefficient for people to read textual descriptions, and pictures tend to be absorbed much more quickly than textual information, and so that's why we've developed so many different visualization tools. And so here's an example, is this diagram is trying to summarize all the regulatory influences on an E. coli gene called ARO-L. And what we're showing here is the transcription process that takes the DNA version of a gene and creates an RNA, and then the translation process that translates the RNA into a protein product. And so what we're seeing is that there are 2 different genes that inhibit the production of ARO-L, the transcription of ARO-L, that is. And those 2 genes are sensitive to metabolites called tryptophan and tyrosine and phenylalanine. Then once the DNA has been transcribed to an RNA and translated into a protein, then magnesium is a known cofactor for that protein. So magnesium has to be present for that protein to be active. So this diagram is generated by our software. There's kind of an overall template diagram that the software can generate, and then based on querying the underlying database for what's known about the regulation of this gene and the gene product, the software overlays these various components of the diagram. So here's another example of a regulation summary diagram. In this case, it's the LRP gene that inhibits the transcription of this gene, and there are 2 small molecules that inhibit the resulting protein product, which requires an iron-sulfur cluster to be active. Here's another version, and that actually looks pretty, that's the same one. And then Let's skip that one. Here's a similar gene, another E. coli gene, but this one has 3 genes that impact its transcription, 2 positively, 1 negatively, and then the RNA that's transcribed, its translation is inhibited by this small RNA. And then the product forms a multimeric complex that's also inhibited by 2 different metabolites. So the idea is that the user hopefully can digest what's known about the regulation of these genes very quickly from these generated diagrams. So the overall approach we take to visualization is, first of all, we really want to separate the user interface from the underlying database. Database representations are designed for computers, not for people. So we've designed these visualizations to transform the information in the database into a form that people can digest very easily. So all our visualizations are automatically generated from the database. And that automatic generation gives us scalability so that people don't spend time creating these diagrams. And it also gives us non-redundancy so that we don't, when the database information changes, we don't want somebody to have to manually redo the diagram. We want the software to automatically generate the diagram. Hopefully these diagrams are intuitive, can be grasped very quickly, but we do have documentation for all the diagrams if the user wants to study them and understand the different visual conventions that we're using. So here's an example of another regulation-related diagram that we call the regulatory overview. And the idea here is to depict this complicated network of interactions between different genes in an organism. So you're actually seeing the entire genome of, well, close to the entire genome of E. coli in this diagram. Every dot here is one E. coli gene. And the genes are organized into these 3 different rings. The inner ring are the global regulator genes of E. coli, where most of these genes regulate tens or hundreds of other genes. The middle ring are other regulator genes that typically will regulate a smaller number of genes. And then the genes along the outside are individual genes that are not regulators. And we've grouped these into these clusters for 2 reasons. Number 1, because that way we can fit them all on the page. And number 2, we've used a biologically meaningful clustering, which is namely each one of these clusters is a set of genes that all respond to the same regulator or set of regulators. And so like this is the set of E. coli genes that all respond to the sigma 70 regulator. I'm not quite familiar with the others. Now the lines in the diagram basically tell us that this gene is regulated by the genes that point to it through one of these arrows. Okay, and if we displayed all of those lines simultaneously, we'd get a hairball diagram that was totally incomprehensible. And so the approach we take is to start out with none of the lines shown, and then interactively the user can click on a gene and say, well, show me everything it regulates. And that way the user can interactively explore what's known about the regulation of all these genes. Now here's a use of the same diagram, but to show a gene expression data set. Now here we don't have any of the lines displayed. We just have our 3 rings of genes. But in this case, we're coloring the 3 rings to show the expression levels that have been measured in a genome-wide transcriptomics experiment, where in one experiment, a biologist is measuring the expression levels of every gene in the organism, perhaps over time or in response to some changing environmental conditions for the organism. And this is one time point, but the diagram is animated so that if there are multiple time points that you have experimental data for, you'll get basically a movie showing the colors changing over those different time points. And then the scale on the left shows us the expression values that the different colors map to. So the orange and red colors are the most highly expressed genes, and the blue and purple are the genes that are not expressed very much. And so one thing you can do with this diagram is you can click on a gene, see what it regulates, and then see to what degree the expression level of the regulator is correlated with the expression levels of all the genes that it regulates. Well, here's another tool that we've developed really recently called the Omics Dashboard. And this tool is designed to help scientists analyze the same type of data that I just showed you, namely transcriptomics data, high throughput measurements of the expression levels of many genes simultaneously. And there's so much data here. Like for E. coli, this means that at each time point, the scientist is measuring about 4,500 genes, their expression levels simultaneously. So if you have 5 time points, you've got 5 times 4,500 numbers to try to digest. It's a lot of data. Now, this same tool can be used not only with gene expression data, but with proteomics data, metabolomics data, reaction flux data. And you can run it through our website. And here's the basic idea, is the tool consists of a set of panels. So each one of these horizontal groupings here is one panel. And then each panel contains a set of plots. Okay, so this is a plot, that's a plot, that's a plot. And you may see a little analogy to a musical score, and there is some similarity here to a musical score. So here's the basic idea, is that every vertical line, so okay, let me start again. Each panel is depicting some major area of cell biology. So these are all the biosynthetic metabolic pathways of the cell that are being depicted in this area. These are all the degradation pathways, energy pathways. These are various cellular processes. And then within each panel, each plot is depicting a sub-area. So this overall panel is all of biosynthesis. This is amino acid biosynthesis. So there are 20 amino acids made by E. coli. There are about 130 genes involved in making those 20 amino acids. And what we're seeing here is all of those 130 genes in the first time point of this experiment, which has, I think, 6 time points. All 130 genes, their expression levels are being plotted across this vertical line. This vertical line represents the first time point in the experiment. The next vertical line is the second time point, third time point, etc. So along each time point, we're plotting all of the expression levels of all the amino acid biosynthetic genes. The fat dots are the averages of those expression levels. And so by letting your eye focus on only the fat dots, you're essentially seeing what's the overall response of this area of the cell's machinery during the course of this experiment. What we can see is that what's happening in this experiment is that E. coli is shifting from growing without oxygen to growing with oxygen. And so one of the pretty simple inferences you can make is the cell's growing faster. Its rate of making almost everything is going up during that transition. So the rate of amino acid biosynthesis overall is going up. And so each other panel depicts some other sub-area of biosynthesis, from nucleotide biosynthesis, fatty acid lipid biosynthesis. I think I'll read these other ones. How do you do that?
Two finger.
How does that work?
On the trackpad, just with 2 fingers.
It's not working for me.
Oh, really? Okay.
That's okay. Well, let's. The next screen actually shows. So the dashboard kind of has 2 goals. One is to let you see what's happening across the entire cell in one screen. So that's why we call it a dashboard, is you can kind of see all the cellular machinery here in one screen and hopefully find out which parts do you want to explore in more detail. So let's say we want to know, well, what's really going on in more detail in amino acid biosynthesis, because there are 130 genes there. And it's hard to, you can't really make out all 130 genes. You can't tell what's going on there. So if the user clicks on that, they get this display. Let's focus on the left side. The left side is now showing essentially the same kind of graphical plot, but now for each individual amino acid biosynthetic pathway separately. So these are all the genes involved in making alanine. And again, they're plotted, we have the same 6 time points. We're plotting that set of genes along each of the vertical lines at each time point, and the big dots are the averages. So we can see alanine biosynthesis is going up. They're almost all going up except for aspartate biosynthesis. Oh, actually, in asparagine biosynthesis, they're both going down for some reason. Cysteine is staying about the same. Glutamine is going down a bit. So that gives us our next level of detail to show what's happening for the pathways for the individual amino acids. Now, if we want to see still another level of detail, let's say we want to know, well, what's really going on in arginine biosynthesis? We can click here, and we get this diagram that shows each gene, each individual gene in arginine biosynthesis and what's happening with it. And we can see that all the genes are behaving essentially the same way. They're all going up as each time point goes on.
That's really amazing. Where did you publish this?
Nucleic acid research just came out pretty recently.
Really awesome.
Thank you. And you can do things like, here are all the genes involved in the TCA cycle. Here they're sorted by genome position, but we can sort them by expression level, by its average expression level that we're sorting them by here. And another set of tools we have let us show the gene expression data across on a pathway diagram. So here we're depicting the TCA cycle as a pathway, and then each one of these little series of boxes shows the expression levels of one gene across all those 6 time points. So this is one gene across all 6 time points. It starts out gray, and then here the colors go. Gray means very little expression, red means lots of expression, so this one is increasing over time. They're almost all increasing over time because this pathway is not used during anaerobic growth, but it's used during aerobic growth. Here we're seeing the organization of these genes in operons, essentially how they're clustered on the DNA. And then here we're showing what the software has done is it's looked across all these genes, it's queried the database to find out what are the regulators that act on all of these genes, and it's plotting the expression levels of all those different regulators on this diagram. Okay, I think I will skip that one. So here's another one of our diagrams. Mentioned this before. This is called the Cellular Overview diagram. And this is a diagram that shows us the full metabolic machinery for an entire organism, as well as the transporters of the organism. These diagrams are automatically generated on an organism-specific basis, so we generate a different one of these diagrams for each of the 11,000 organisms in BioCyc. And these diagrams are zoomable and queryable, kind of similar to Google Maps. So this is the lowest resolution view of the diagram. This shows us the human metabolic map. And the way the diagram's organized is we have all the biosynthetic pathways on the left, the degradation pathways here, energy pathways here, and these are individual metabolic reactions that are not assigned to a particular pathway. And then transporters are along the outside. We haven't populated this with many human transporters yet. And then each dot is a single metabolite, and each line is a single metabolic reaction that interconverts these metabolites. Here's the E. coli version of the diagram. Same basic organization, but the details differ. E. coli and humans actually share a lot of metabolic pathways in common, but each one has a bunch of pathways that the other one does not have. And that's actually one operation that you can do in the software is you can say, color me in red every reaction line that's shared between humans and E. coli. So you can see what things are shared between the two and what things are unique to E. coli. And so you can zoom into the diagram and show, here we're zooming in more and more to see a higher resolution view of the diagram, until finally you get a version of the diagram that's basically a metabolic wall chart that you can print out on a large printer and put on your wall. Now here we're using the same diagram to paint gene expression data onto. Right, so with Google Maps, you can show traffic data on a map diagram to show which areas of the highway are moving at different speeds. So here we're showing which reactions within the network have gene expression levels of different amounts, so that you can see the relative activation levels of different parts of the metabolic map. So this is yet another tool for visualizing gene expression data by showing kind of in a pathway context which parts of the network have different activation levels. And you can also use this in an animated form to kind of cycle through a series of time points and see how expression levels change for a given region of the map. And then you can click on a given reaction to say, well, show me in detail what's happening for that reaction over the course of time. And here's metabolomics data painted onto the same diagram. Metabolomics data are basically high throughput measurements of metabolites. And so rather than coloring the reactions, here we're coloring the metabolites to show what levels are present for them. And we can also do the same type of thing on an individual pathway diagram. So here we've taken an individual pathway diagram, and we're seeing for each step in the pathway, what are the expression levels of the genes at that step. And here we have what we call a pathway collage diagram. So previously we saw the entire set of pathways of the organism versus one pathway for the organism. And what users told us is, well, this is kind of too much information and this is not enough information. Sometimes I want to look at just a small number of pathways that are linked together. And so we developed a tool that lets the user say, okay, here are my 6 pathways I'm interested in. And the software will lay them all out and let the user move them around relative to one another and create this personalized diagram that shows a set of pathways of interest.
Is the data points on how much each thing's expressed, is that like aggregated across everything that's been put in the database, or is it like the result from during one experiment?
It's the result from one experiment. So on E. coli, I don't even know how many of these experiments have been done around the world for E. coli. It's probably hundreds. And so the idea is that the user takes their data set that they've just generated and they can upload it into the tool and visualize it. Or another thing they can do is there are other databases around the web that contain kind of archives of these different data sets. And so I might be interested today in finding some data set that Bodan published 5 years ago, and I can just pull that up and put that into the same tool. Now here's another visualization we developed. It's called the Genome Overview. The idea here is to show the entire genome of an organism in one screen. And so this is kind of organized like you read a book, right? You read across the top row, and then the next row continues it. So this is essentially the full DNA of the organism, kind of broken out in this book order, where each triangle is one gene. Each triangle here is one E. coli gene. So we're seeing 4,500 genes here. The triangles are all the same size, so the genes are not to scale, because different genes are different lengths. But we've chosen to get rid of the scaling aspects of genes. The colors indicate operon membership. So a block of adjacent genes in the same color are part of one operon. And then The left-right directionality of the triangles indicates transcription direction, and then the up-down indicates protein genes versus RNA genes. So the protein genes are up-pointing arrows, and the RNA genes are down-pointing arrows. And we can create a genome poster out of this diagram. We can also visualize gene expression data onto this diagram. So here, the color scheme has just shifted. So instead of having the colors represent operons here, the colors are representing expression levels of individual genes. So you can see which parts of the chromosome are highly expressed, which clusters of genes have similar expression levels. So it's a genome position-based view of gene expression data. And that's the end of my talk. Let me acknowledge my funding source, which is NIH, and various collaborators on my team who did both the curation and the programming behind these different tools. And happy to take more questions if you have them.
So you said that you've had bioengineers use this to be, if we modify this pathway, like what are some downstream effects? Have there been any like pathogen host interaction with people using this database? So if we add this or this pathogen protein that's injected during an infection, how does that modify these pathways?
Yeah, there probably are. Scientists in those areas are probably using the databases. I don't know in detail what they're doing. We don't have those kinds of interactions really highlighted in our databases between pathogen gene products and hosts. Those genes are there, but we don't kind of highlight them the same way we do the metabolic genes. But yeah, that's an important area that maybe we'll address one of these days in this more specialized way.
Yeah, because like specific defense compounds are turned on during these, so would their like metabolic pathways be in this data set, just not necessarily tagged as like defense metabolic pathways?
Then, yeah, I mean that's a good example where that's a good example of an interaction that is metabolism based. And so our site right now only has one plant, which is Arabidopsis, and I don't know how many pathogen defense pathways we have in the database, but there could be some.
Can you talk a little bit about kind of the flow between using Common Lisp and JavaScript and some of the diagrams that you showed up here today? Like some of them look very, very impressive, and I was just wondering, like one thing that comes to mind, of course, a lot of people are interested in interactive visualization these days, so D3JS is really popular, and so one question is, have you tried using that programming language as part of your BioCyc framework? And another question is kind of what is the kind of split between what you use Common Lisp for this research, and where do you just do JavaScript, and then where do you combine them? It's kind of a general open-ended question.
Yeah. So Common Lisp is used basically on the back-end server side. It's used for our underlying object database system. It's used for lots of different application code that computes biology-specific things, like for example, the code that translates DNA to RNA, or transcribes DNA to RNA, translates RNA to protein. That's all implemented in Lisp. All the metabolic reasoning code that does things like, you can ask questions like, given a gene, what are all the pathways the gene's in? Given a pathway, what are all the genes in the pathway? The metabolic root search, that's all programmed in Common Lisp. Basically, everything's in Common Lisp. Even much of the visualization code is written in Common Lisp. Now, the JavaScript code, let's see. That's actually been changing very recently. One of the problems with our Common Lisp visualization code is that it relies on a whole Common Lisp graphic system called CLIM, C-L-I-M, that's now very old, very antiquated. The fonts look old. The graphics they generate are not aliased, anti-aliased rather, so they look really jaggy. So over the last year, what we've been doing is to come up with an automatic translation from the Common Lisp graphics into JavaScript. And so a lot of our diagrams now in the latest version of our software, which we just released last week, are doing that automatic translation. And so now we have modern fonts in our diagrams, and now we have anti-aliased graphics that look much cleaner. There's still a few exceptions. One example being this diagram is still the old version. And the thing about this diagram is when you are zooming it the way I showed, you use this little control on the left side, similar to Google Maps. And basically what it's doing is we have pre-generated sets of images for the different levels, and the JavaScript is going to the server and retrieving different images depending on what zoom level you're at and where you are in the diagram. Now what we have now is, what we've done is we've created this system that automatically translates the Lisp graphics into a really compact graphical description that's essentially a series of line drawings, circle operations, text drawing operations, that's sent from the Lisp server to the web client, where there's a JavaScript drawing engine that basically interprets all those graphics and draws them on a JavaScript canvas. And it can draw them fast enough that we can now zoom this diagram in real time, so that as you're kind of scrolling your mouse wheel, the size is changing. It's drawing the entire, it can redraw the entire diagram in like 50 milliseconds. Wow. So, and it decides, you know, at what magnification do I start showing the fonts versus I just omit the fonts. So there's some nice semantic zooming in there as well. So, yeah, and the advantage to doing that is we didn't have to rewrite all the Common Lisp graphics that we have. We reuse all that code, and we're automatically generating the JavaScript graphics. Some other places we use JavaScript are like, I think one of the nice tools we have is one of the first things you do when you go to biocyc.org is you want to select which organism you're working with. Okay, there are 11,000 organisms. Well, we have this in the upper right-hand corner. It says select organism. You click on that and you get this dialog with like 4 alternative tabs in it that lets you enter organisms by name, or there's a taxonomy browser. What else? There's actually one of those tabs lets you select organisms according to phenotypic properties. And that's all written in JavaScript. So essentially things that we want to operate on the browser side are written in JavaScript. And we're kind of making a transition from kind of the older server-based way of doing things to try to do more and more things in the browser so it's faster and offloads the server. So kind of a long-winded answer, but...
Oh, very nice.
Other questions? Yeah?
I'm curious what Common Lisp implementation you use.
We use the Franz product, and we've been really happy with that. They have...
When people license your code, do they license Franz as well?
They do, and so the way that works is we worked on an agreement with Franz years ago that we distribute the software for free to academics. So that's, most of those 8,000 licensees are academics. They get it for free from us, and because we don't charge anything to them, Franz is willing to let us distribute the Common Lisp runtime for free. There are roughly a dozen commercial subscribers that have bought the software, and so we charge them money for it, and then Franz gets a share of those license fees. So that's how that works. So yeah, they've been nice in working with us to, because the government likes us to give it for free. The government agencies that fund us like us to give the software for free to academics. That's kind of the standard in the bioinformatics world.
Are there any predictive capabilities of this, or are the changes in flux like that dashboard you're showing going into kind of experiment with data that's already been generated?
Well, one example is the metabolic modeling stuff, which I didn't really spend much time on in this talk. But when you have a metabolic model, it's basically a steady state model where the user can specify what set of nutrients the organism's growing on. And the model will calculate does the organism grow or not on that set of nutrients, and when it is growing, kind of what are the flux rates through all the different reactions in the metabolic network. And so that's predictive in that it's predicting growth or no growth. It's predicting actually what the growth rate is, and it's predicting what are these different flux values. Another thing you can use it for is you can do computational gene knockouts. So you can, the models run very fast. They run in under a second. So in like under 10 minutes, you can knock out all different genes in E. coli, run the metabolic model with essentially with the reactions that those genes catalyze disabled, and then see if the cell is predicted to grow or not. And then you can compare that with experimental data to find out. That's one of the ways that people validate the models and assess how accurate the models are, is by comparing the computational predictions of growth or no growth against experimental data. And so if you go to for 3 of the organisms that we have metabolic models. One is E. coli, and so through the website, you can go and run the metabolic model online.
I guess in humans, do they do that?
We don't have a We don't have a human model right now. There's another group that has a human metabolic model. So if you did a Google search for like human metabolic model Recon 2, it's called reconstruction Recon 2. Yeah, and there are other models out there too for humans, but yeah, they have a metabolic model they've spent a lot of time developing, and there are all sorts of uses for the model. I mean, one of the really interesting ones is to look at cancer cells where they might have different metabolic genes inactivated and try to figure out how does the metabolism of cancer cells differ from normal human cells. One of the things I'd like to do really is to have metabolic models for many different human cell types and be able to compare what metabolism is active in different human cell types. Funding problem there, so.
Human Cell Atlas is really big on that right now. So the large scale initiative that just started, I think it was in October of last year.
Right.
They're doing a lot of work in that direction, so that's a very good funding source if you want.
Yeah. I should have submitted something to
A lot of people here at Stanford, and then a few folks at the Broad, MIT and Harvard, and then a few other groups around on the nation, and also internationally as well.
Yeah.
So what you just described, I think, is something that would really resonate with them.
Yeah. But yeah, I think thinking about personalized medicine, I think one day we're going to have personalized metabolic models for different people coming from their genome sequences where we can make predictions about how their metabolism differs.
How to find the right drugs to treat their
Yeah, because a lot of diseases are metabolism related.
Yep.
Aging, obesity, diabetes, the list goes on and on.
Right. How to minimize side effects by knowing them not all have the personal features. A lot of time for that.
Well, thanks everybody for coming.
Thank you.
Welcome.